Okay.
Greetings, everyone, and welcome to today's episode of 17 Minutes of Science. I'm Janice Weeks, the co-founder and chief global health officer of In Vivo Biosystems, and I have a background in neuroscience and drug discovery. And we're happy to have all of you here with us today. It's my pleasure to introduce today's guest, Kyle Galford, and he will be speaking about tiny worms with a big impact, using C. elegans for toxicology studies. So let me start by just saying a little bit about Kyle. What led to Kyle's appearance on 17 Minutes of Science was a paper he published recently entitled "The FDA-approved drugs ticlopidine, sertaconazole, and dexlansoprazole can cause morphological changes in C. elegans." And he did this work while he was an undergraduate at University of Maryland, College Park, in the lab of Antony Jose, who's the other co-author on this paper. And when we contacted Professor Jose as a potential speaker, he generously suggested we feature Kyle instead because he's the one that actually did the work. And that's a sign of a great mentor. So kudos to Professor Jose. So in this study, Kyle screened a library of FDA-approved drugs looking for morphological effects or lethality in C. elegans. And the recent paper describes interesting results for 3 of the drugs. And after graduating in cell biology and genetics at University of Maryland, Kyle took a position at the biotech company Novogene, where he is now. Okay, I'm going to start my timer for 17 minutes. Great. And also, if you have a question for Kyle as we go along, please type it as a comment on Facebook. So I want to start out with your research as an undergraduate. And this interests me because so many scientists in academia and industry, myself included, first got into lab research while an undergraduate. So can you tell us a little bit about how you began doing undergraduate research and what the experience was like?
Sure. And thank you very much for the introduction and for having me on too. So yeah, I noticed when I was at University of Maryland, so I had always wanted to get into science and biology, and that's what decided my degree. And then I noticed that all of my peers were starting to get into research, and I started asking myself what I'm interested in. And again and again, the epigenetics kept coming up. So I was looking for exciting research in the field of epigenetics. And that brought me to something called the Transgenerational Brain Initiative at University of Maryland. So this is a research stream for incoming students that was started based on the finding that double-stranded RNA expressed in C. elegans could silence a gene of matching sequence in the germline and actually persist for into the next generation. So this is the idea that, potentially experience could be transmitted from parent to progeny. And so I reached out based on that, and I wanted to get involved in that stream. And Antony actually had a different project in mind, where he had this novel transgene that they found in C. elegans that, when inherited through a male worm into the progeny, is silenced stably for many generations. And the transgene would express GFP and mCherry if it was unsilenced. So now we had a excellent organism to test the effects of compounds on transgenerational gene expression and maintenance of expression states across generations. So that's really how I got into research, and specifically this project, that's how it came to me. And that's where the work started.
And that the whole transgenerational effect is so important. And, a good example is thalidomide, obviously, where if it was given to pregnant women, it could cause problems in the pregnant woman, but also in their female, or I think both sexes, actually, progeny. So that's the transgenerational drug effect. So that's one thing that's really cool about the work that you've been involved in. So how did you choose to use C. elegans for your research? And I know it was probably, that's what they worked on in the lab, but can you tell us a little bit about the utility of C. elegans for toxicology work?
Yeah, definitely. So the lab was using C. elegans because of the features and its powerful features, and these are the same features. Is the reason that they ended up in this experiment too, and being our model of choice. So C. elegans is extremely well characterized. It was the first multicellular organism to have a complete genome sequence drafted. Um, it's also very small and tractable and easy to handle in the lab. It has an extremely short generation time. So when you're talking about transgenerational effects, it can go from adult from egg to laying eggs in 3 days. And that's extremely powerful. And they can have up around 300 progeny is the typical brood size. So you have a large N coming very quickly when you're talking about transgenerational studies. Additionally, its utility in a toxicology screen, and one where we're looking at fluorescent reporters as well, it's transparent. So if we were to disrupt the silencing with a compound in our screen, we could see the expression of GFP and M-cherry readily. And we could also see any morphological changes, which we did see very easily looking under even a dissecting microscope. And then, moreover, it's also a multicellular organism with many tissues and organs. So these are good models to use for an in vivo system, because you're not looking at, say, a cell line on a plate. You're not looking at a reaction in a test tube. You're looking at a full organism and the effect of whatever you're looking at on a full functioning system.
And was that the first time you'd worked on C. elegans?
This was my first time working with C. elegans, and I had only heard about C. elegans before about once or twice in my biology classes. So I was very surprised to hear how many genetic tools were available for the worm and just how powerful it is, and kind of surprised that I didn't hear about it more before.
Well, that's too bad, but you saw the light eventually.
Yeah.
Yeah. So in the paper I mentioned, you and your advisor screened a large library of FDA-approved drugs. And there's a couple of approaches to discovering new therapeutics. One is, say, trying to repurpose ones that have already been through FDA review and their safety profiles are known. And the other is to try to find new drugs de novo, say by screening natural products. So could you say a little bit about the value of repurposing drugs and screening existing libraries?
Sure, definitely. So there are those two routes. You can either come up with a new novel drug that hits either a new target or a known target, but that takes a long time and it takes a lot of money. And sometimes those are luxuries that we don't have, or the need to alleviate the suffering caused by a particular disease, and in a lot of cases, very rare diseases and orphan diseases. It could be beneficial to kind of truncate that amount of time and the cost by looking towards compounds that we already know about. So with the advent of bioinformatics and large data sets now available, we can look at the drugs and the targets that are already known to us and save on that time and that money, and use something where we already understand the toxicological profile of these drugs and kind of skip some of the early chemistry research steps that take up a lot of time, energy, and cost, and kind of move towards repurposing that drug for a new indication. Yeah.
Yeah, because as you said, the route to a new drug or a new vaccine, which we're hearing about, there's a lot of steps and a lot of expense involved. So yeah, repurposing is a really valuable approach. So then, speaking of the drug screen you did, you settled in this paper, you focused on those 3 particular drugs. So could you tell us what was interesting about those 3 and why you focused on them? Because they're very different drugs, different modes of action and targets.
Yeah, definitely. So we had been looking for drugs that would interrupt this transgenerational silencing, but we didn't exactly see that. But when we looked back through our pictures, we found a few drugs that did cause very distinct changes that you could even see under the fluorescent microscope. Now, this was autofluorescence. But for, say, ticlopidine and sertaconazole, they caused very noticeable morphological changes in the pharynx. And specifically, we think this is the marginal cells of the pharynx that are kind of collecting these compounds in the pharynx of the worm, and in some cases causing lethality. And then in dexlansoprazole, when we looked back through the images, we found a molting defect. So that was very striking, because that's a very conserved process in these nematodes, and it uses a lot of genes that are actually found and pathways that are actually found in humans. So when we saw these, it wasn't necessarily that we had chosen to target these and to look at these, but they are something interesting that we found afterwards. Yeah.
So then you've moved on to a new job. So how is the lab, the Joe's lab, going to move forward, or what's next with this general research trajectory?
Sure. So now the lab wasn't intending to do this, I don't think, before the paper, but now they found themselves as drug researchers. So because these are FDA approved, there is kind of an ingrained significance in the finding, just because, well, now we see that there must be some sort of target and some sort of effect. So to investigate that is the next step. So we'll be looking for requirements for drug uptake, receptors, and the different pathways that are affected by these drugs. So probably is some of those more pharmacological studies coming from the Joe's lab in the future, hopefully.
And again, to circle back to the transgenerational aspect, I mean, that's so important, and I think often not investigated for a lot of drugs. I mean, especially for humans, you know, to go from one generation to the next takes a lot of time. So I'm expecting lots of exciting things to come out of that line of work.
Yeah, this was definitely, because C. elegans is so useful and can be modified so easily, even in this, in our research, even though we didn't find a hit on this transgenerational, looking for something that would disrupt the transgenerational silencing, we still developed a worm that is more sensitive to compounds in their in liquid medium. And they still could be used to find something that would disrupt that silencing. So there is a tool out there now that exists that came out of this, and that is pretty exciting.
And there's so many libraries. I mean, you looked at that FDA library, and there's small molecule libraries, there's natural product libraries. So the fact that you now have this tool, transgenic C. elegans, to facilitate looking for transgenerational effects, I think is really a big contribution to the field.
Yeah, so definitely.
Oh.
No, sorry, go ahead.
Okay, let me go on. We have about 5 minutes left. So is there anything that surprised you about this study, or is anything you weren't expecting that popped out of it?
So actually, what was the most exciting is that we, prior to testing the FDA-approved compound libraries, we actually tested a library of epigenetic inhibitors. And we were actually really surprised to see that nothing hit. So just, I mean, not that we would necessarily expect it, but these have been demonstrated to interrupt things that we thought might be taking part in the silencing. So, the fact that we didn't get a hit from that was surprising. And the fact that we found a molting defect was extremely surprising, I'd say. That came as kind of a shock later, looking through the images afterwards.
Yeah, I used to work on insect molting, and that's such an interesting multi-step, complicated part of the animal's life cycle. So yeah, it affects a lot of pathways.
Yeah, and that also, I suppose another interesting finding and surprising was that dexlansoprazole doesn't have that effect of disrupting molting unless you expose it from the early larval stages. Once it's gone through its early molts, you don't see the effect if you're exposing anything, I think, L4 and above. And beyond that. So that's also pretty interesting. So it has kind of a developmental factor involved as well.
Definitely. So let me switch gears a little. So you were a university student, you graduated, and now you've transitioned from academia to industry. So in your experience so far, how do those two environments compare, and what's it been like?
Yeah, so I made the choice to go from kind of academia to industry, and that's I noticed that in academia you get the depth. So I spent a long time reading a lot of worm papers, but
Uh,
yeah, and in industry now though I get the benefit of kind of getting the breadth now, where I'm looking at or I'm hearing the exciting cliff notes of new projects from
mhm.
all over my territory. So I'm a little part of a lot of really cool research that's going on now. Um, and there's a lot of excitement with that, just like there was a lot of excitement getting the paper published and going through um, all of the publications and learning to get that paper published um,
Yeah.
for my own
So we
research.
have a question that came in from our audience. Are you still working with C. elegans in your daily job? And I think the answer is no. But how much has what you learned about C. elegans helped you in your current position?
So I think it's given me a perspective, just research in general. When researchers are saying that they need to prepare samples, I understand now kind of all of the labor that's involved. And when you have to run these screens, I understand how much work that is. It's also given me, working with C. elegans has given me a chance to look at a lot of different pathways that are conserved across many organisms, including humans. So even if I'm not directly familiar with a particular human or mouse pathway, I know that more often than not, there's something kind of shared in C. elegans that I might have heard of before.
Great. Let's see, we have in a minute and a half. So one more quick question. So you're a young or new generation researcher. So what do you see about the future of research going forward? Different or new ways to conduct research? We've seen some of that in the COVID pandemic, but what's your vision going forward about the directions?
Yeah, I think things are going to become more and more remote and quick and technological. So, research can be done now using data sets that are already online. So I think kind of pulling together these large data sets from large screens and sequencing experiments and so on, that'll yield a lot of insights that maybe we didn't have in the past, we didn't have access to.
Yeah,
Yeah, I think large collaborations with big data sets is kind of where it's going.
right. I mean, science has moved so much more into bigger teams, both in the US and around the world. So, and I think the current situation has just promoted that even more since we can't all go to the lab as much
Yeah,
anymore.
yeah, definitely. It's amazing how quickly a lot of research pivoted when this all started, and just being able to see that in my job now.
Yeah. And I agree, bioinformatics and data mining, all you need a computer and a source of data. So it's something we can all do at home, even if we can't go in the lab and pipette things.
Yeah, definitely.
So, okay. Well, we're just about out of time. It's going to go off in just a minute or in 7 seconds. So, Kyle, let me thank you again for being on our show with us and taking the time of your busy day to do this. And we wish you all the best in your current career and working with different customers through Novogene. And for all of you out there watching, thanks for spending this bit of time with us. And we look forward to seeing you again next week for another episode of 17 Minutes of Science.